So a couple months ago, I bought a can of tomatoes. And I noticed there was this very interesting label on there. Non-BPA lining. Non-BPA? What's this BPA that they're taking out of my canned tomatoes? If they're taking it out, why are they taking it out? Does that mean that this BPA thing is bad? Unfortunately, the answer to that question is yes. This BPA thing is bad. And so what our project is focused on is remediating this BPA thing from our wastewater by biological means. And so this is what I
and Izzy
will be presenting today. So BPA, it is categorized as a pollutant of emerging concern because, well, emerging being the sense that it's used in the production of modern plastics. You've got canned goods, ski goggles, plastic bottles, toys, you name it, it might be in there. The concern part is in regards to the fact that it has a similar chemical structure to estrogen. And because of this, it disrupts regulatory hormone systems in the body. So you have a cell, and this is a cell membrane. You got receptors on the cell membrane, and normally what happens is a natural hormone will bind to this receptor and elicit a response from the cell. But the thing is, an endocrine disruptor, which is what BPA is, does that exact same thing because it mimics that same structure. And so that produces a plethora of detrimental effects. And so when BPA gets into the environment, we can see its effects happening in real time. Here's a fish, how you normally see it when it's out in the environment, and you notice the arrow is pointing to its lower abdomen. This is going to be very important later on. And so this is a fish with low BPA in it. This lower abdomen, okay, it's growing a little bit. And then finally, high BPA, in which this lower abdomen has protruded substantially.
So where does BPA even come from? Well, BPA is very prevalent in our society because it can be found in a multitude of plastic products. For example, it can be found in children's toys, plastic bottles, discs, and even food packaging. So how much BPA is not removed? Well, BPA enters our wastewater when the product is thrown away, represented by the black dot. This BPA then goes into a wastewater treatment facility, where processes targeted to remove pollutants remove the BPA. However, 2 to 9% seeps through because it either stays in the sludge or heads to the ocean, heading to the environment. The sludge is a bacteria sludge that wastewater treatment facilities use to break down the harsh bonds in BPA. When it's done and fully does its service, it's head into the environment. Looking at current removal methods. Firstly, there's adsorption. Adsorption is like a sponge. It removes 66 to 99% of BPA, depending on the material used. However, some drawbacks to this process is that it's inflexible, meaning that it can only work under specific conditions. Also, it has a limited removal capacity. And so once that threshold is met, BPA remains in the water. There's also advanced oxidation processes, which removes 90% under optimized conditions. A drawback to this, though, is that harmful byproducts pursue. The advanced oxidation processes involve introducing a harsh chemical which breaks down the strong bonds in BPA. Sometimes these byproducts, though, can be more harmful than the BPA itself. Also, advanced oxidation processes require a lot of extensive monitoring. So we need new ways to remove BPA. So what about microalgae? Well, first, what even is microalgae? Microalgae is a smaller version of algae. Looking at the picture on the left, you can see what you would regularly refer to as an algae species, and it's usually bigger and found in oceans. Looking at the picture on the right, you can see microalgae, which is one cell or one unicellular organism. So why are we using microalgae as a removal tool of BPA? Well, it's scalable for these wastewater treatment facilities. They can either increase the amount of microalgae they use, depending on the amount of pollutants in the wastewater. Also, microalgae has a high tolerance to different salt concentrations, pHs, and other pollutants. And finally, the products of microalgae are reusable. For example, microalgae produces a lot of oils, which can be harvested as renewable energy. So, could microalgae be the key to efficient BPA removal?
If we use the microalgae, how would they even remediate it? Okay, so we got our first method here, adsorption, in which the BPA sticks to the outer membrane of the microalgae cell. Next, we have bioaccumulation, in which the cell takes in the microalgae and stores it inside of the cell. And next would be biodegradation, where it's like accumulation, except that the cell takes in the BPA and uses enzymes to degrade it. So we've got a hypothesis, we've got a theory. We put the algae in the wastewater treatment plant, it does its remediation, boom, bada bing, we're done. No, we're not. We were discussing about in what kind of forms would be the most ideal to put it in a wastewater treatment plant. We've got free-flowing, planktonic. More surface area, that means higher rate of reaction. Okay, that sounds feasible, but we have another option. We've got immobilized beaded form, in which you have millions of microalgae inside of a little capsule, so that when it's done remediating, you can just take it out of the process, you can scoop it out easier. We chose to go forward with the beaded microalgae. And so there were two main species that we were looking at. The first was Chlorella pyrenodosa, which has extensive research on its remedial properties of pollutants. So it's well known to remediate different kinds of pollutants, including BPA. Next is Chlamydomonas pacifica, which was only discovered 5 years ago. So there's still a lot to uncover about this microalgae species. Microalgae might be cheaper, might be better for the environment, but there's still a lot of questions that we have to answer. First, what light intensity is the most optimal for remediation? Second, what's a good pH range that would be the most optimal? And third, what species would remediate better, or in what form? Would free-flowing remediate better, or beaded form? Here's what we know today. In terms of Chlorella pyrenodosa, in the literature reviews, it has been shown to remove about half to 95% of BPA over the course of almost 2 weeks. And with Chlamydomonas pacifica, although there is no direct evidence that it can remediate BPA, it has been tested with sister strains to the result of 1/4 removed around after 10 days. Here's the specific research questions that we have devised. First, does the bead form remove pollutants? Second, which microalgae types perform better? Third, how does high pH affect remedial rates of BPA? And fourth, how can we track BPA removal so easily?
Looking at our experimental setup, we used 2 plate shakers as shown by the image, which would rotate our samples so that all the wastewater could interact with all of the beads. This was so that we could remove as much BPA as possible.
So here's our preliminary results. Does the bead form remove pollutants? Yes, but there's a catch to it, which we will get later on. And which microalgae types perform better? So we looked at 3 bead types here in our experimental study. We have just Chlorella pyrenodosa in itself, just one species in the bead. Then we had Chlamydomonas, just itself in the bead. And we had a co-culture, which had both Chlamydomonas and Chlorella pyrenodosa in it, in a singular bead. And the next question, how does high pH affect the remediation rate of BPA? Okay, so we looked at 2 main pH ranges here. We had the pH 7 through 8, which is kind of what basic water is, very neutral pH. So we would say, okay, we're gonna put Chlorella in there. But for pH 10, BPA is easier to remove in this basic pH. So we're like, Chlamydomonas can also thrive in this pH, so we're gonna stick some Chlamydomonas in there as well. And the next question, how are we able to track BPA removal so easily? We've got 2 ways to detect BPA. One is $50 to $150 per injected sample. It takes weeks to get results, and we don't get to see it in real time. And this is high performance liquid chromatography, which does give us results. It is legit, but we don't get to operate it in real time, and it is pretty expensive. Whereas this next method is $15 per sample. Only takes hours to get results, and we get to operate it in real time. And so this is ELISA, or the enzyme-linked immunosorbent assay, that we got to operate in real time. So here's our adventures with it, because there were certainly some ups and downs with it. In order to find the concentrations of BPA in our experiments, we had to derive it using known concentrations that were already provided to us, which is called standardizing. So we created these standards which had known concentrations already in them. This sort of standardizing, or standard curve, which we will get into, sort of acts as a ruler or a scale bar. in which everything we measure is in relation to this ruler and scale bar. This is a standard curve. This blue line is the standards that the kit provided to us, and the red was the standards that we made our own. And so if the concentrations are right, then we get a straight line. So it's like, okay, we can pipette, we got good technique, we can do all of that. If we are accurate, we are able to derive BPA concentrations from absorbance values we get from ELISA. So absorbance in the y-axis and BPA concentration in the x. Here's how we tested the effect of the high pH on BPA removal. So there were three main groups that we looked at. The first was the pH 7, which we discussed had the chlorella. And then we had a group of pH 10. But this is with a carb bicarbonate buffer. Now a buffer is just what keeps the pH in the range. So it kept the pH at pH 10. But if you notice, it dissolved the membrane of the beads. So now this algae is free-flowing and planktonic in this flask. And so the third group was the CAPS buffer, which was pH 10.5, but it didn't dissolve the membrane this time. And so here, with the CAPS buffer, here's the results after 1 day. Wastewater, 28% increase. Empty beads, 63% decrease. Pacifica, 2,000% increase. And the co-culture, 37,000% increase. How did we get here? Like, did somebody sneak into the lab and started spiking the flask with BPA? Like, what happened? Did we have good technique? Did we have bad technique? We had good technique, though. You see, these were all the thoughts that were permeating in our minds when we saw these results. So there's a lot of questions left unanswered and a lot of anomalies that we had to deal with. What we learned from this was, okay, 1 day, it's 435 nanograms per liter. The next, it's 162,000. And it's just a continuous cycle of roadblocks after anomalies after roadblocks. And yes, it's very demoralizing that we don't get the answers we want. But if we paint this in perspective, this is the first time, or rather one of the few times now, currently, that we are studying remediation with beads. Because most of the stuff is with free-flowing planktonic. So this is novel research that we are participating in. And it's the first attempt, or one of the few attempts, that we use Chlamydomonas pacifica to remediate these kinds of pollutants. And it's one of the few attempts that we've tried to track BPA with ELISA. So in the grand scheme of research, this just, in reality, puts this whole thing forward. Because we are finding novel information every time we run this.
Going on to lessons that we learned. We learned that mixing matters. If you look at the picture on the left, you can see a yellow solution as well as a blue solution in our wells. This was because we did not thoroughly mix our solutions and had to put it in a plate shaker as a result. Also, measuring matters. We were working with really small quantities, and so ensuring good lab technique was important. Thirdly, we learned that teamwork matters. In the lab, each of our teammates had a job, and we had to trust each other to do that job to the best of our capabilities. And finally, we learned that ELISA is very sensitive. But even though it is sensitive and we didn't get the results that we wanted, it does not define ourselves as researchers. Some future studies. If we had more time at the lab, we would want to see if we can further clean the samples before using ELISA to ensure the best results. Also, we would want to test how does non-beaded microalgae work? And finally, we would want to verify the BPA concentrations with HPLC. Some insights. Some of my insights that I learned is that pollutants can be invisible. Even though you don't see a plastic bag in the ocean or particles does not mean it's clean. Also, remember to be careful when using ELISA. Just because you have good teamwork and good technique does not always ensure the best results. Thirdly, make sure to use research. Just because a research paper is lengthy, has hard vocabulary, or has struggle accessibility, make sure to use it. And finally, invest in yourself and your research pursuits. If you're curious about something, pursue it. So how can you participate? Some tips on avoiding BPA. Look for BPA-free signs when you're shopping, particularly for plastic products. Also, use glass and stainless steel instead of plastic when applicable. Thirdly, do not heat plastic containers in the microwave or mix plastic with anything that's hot. Fourthly, discard damaged plastic goods. And finally, avoid plastic line or BPA line cans.
So we would obviously like to thank our parents, our families, to those who drove hours to get here and those who didn't. We appreciate your contributions nonetheless. We would also like to thank Dr. Liz, of course, and Mr. Manji. Our TA, Kira, our awesome, super duper, we call her a rabbit. The R in Kira stands for rabbit. That's how you know how much we like her, because we have those inside jokes. Our lab partners, Ricky and Yana, absolute goats, absolute real ones. And the past Project 4 students who were able to pioneer and able to set up the foundation for us to work with microalgae. And also the Catalyst Research Program, and by extension, Dr. Liz, and also Oakcrest Institute of Science, who lend their space for us to pursue our scientific inquiries.